This video is produced by Sammy and
Kevin Burgess
for by Inquisition Press to support the workbook Sophomore Organic Chemistry 2 by Inquisition, available from www.byinquisition.org.
What I'd like to talk about now with you is Chapter 16, Peptides and Proteins. Sammy, what's the symbol for gly?
GLY.
And where do we draw the peptide N-terminus? Do we draw it on the left or on the right?
You draw it on the left.
Good. Let's start by drawing triglycine. The symbol GLY refers to NHCH2CO. If someone drew gly, gly, gly as those three-letter codes, it doesn't really make sense because that doesn't specify what's on the N and C termini. The peptide N-terminus is conventionally drawn on the left-hand side, and if it's a free amine, then we would state hydrogen, gly, 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 and OH if it's a free carboxylic acid on the C-terminus. Within each amino acid of a peptide or a protein, there are certain terms that we use to describe the dihedral angles. Phi denotes the angle between the NH and the C-alpha substituent. Psi denotes the angle between the C-alpha and the CO. And omega denotes the angle between the CO and the NH. But we know from resonance that amides are flat. So really, in peptides and proteins, the amide bond is constrained to be either cis or trans, and it's the trans form that's the most prevalent. To experimentally determine the sequence of amino acids, we use the Edman degradation. That involves reacting the N-terminus with phenyl isothiocyanate, giving a thiourea. The thiourea doesn't react further until acid is added to protonate the carbonyl, cause cyclization via attack of the sulfur on the carbonyl carbon, and then hydrolysis to liberate, after rearrangement, a thiohydantoin. You see, the process is stepwise. It doesn't proceed until acid is added. So after acid is added, then another molecule of phenyl isothiocyanate can be added to the N-terminus, and the sequence repeats. We go through cycles of addition of phenyl isothiocyanate, and then protonation generates a heterocycle. Each thiohydantoin liberated in the Edman degradation has a retention time which is characteristic of the amino acid side chain that it carries. An alternative to determine the primary sequence of a peptide is to cleave the peptide into smaller fragments and analyze these via mass spectrometry. Here I have a little picture of a protein or a peptide, and it's being cleaved by trypsin. And why does this lysine look so nervous?
Because trypsin cleaves the C-terminal side of lysine.
And what about the arginine here? He doesn't look happy.
Yeah, and arginine as well.
So if a peptide is treated with trypsin, trypsin only cleaves the sequence on the C-terminal side of lysine and arginine. The smaller peptide fragments can then be analyzed by MS and characterized. Alternatively, if the peptide is treated with chymotrypsin, that cleaves only on the C-terminal sides of amino acids that have side chains containing a 6-membrane, whereas elastase cleaves on the C-terminal sides of the smallest amino acids, gly and ala. Each of these enzymatic cleavages gives smaller peptide fragments with overlapping sequence information, and that enables the whole primary sequence to be elucidated. There are also chemical methods that can be used to cleave peptides into smaller fragments so that they can be characterized. One of these is cyanogen bromide, which selectively cleaves methionine residues. One of the beautiful things about peptides is that as they grow, They hydrogen bond more between different residues to give secondary structures. It's quite easy to look at pictures of protein depicted by ribbon diagrams and pick out some of these secondary structures. To reduce the number of conformations available to a peptide, it is possible to make constraints on the sequence. The most obvious constraint is just to join the C and the N termini together to form a loop, a cyclic peptide. Another way to cyclize peptides is to take two cysteine residues and oxidize them to form a disulfide. If there's more than one cysteine residue in a peptide, then you can form two disulfide bonds, and in that case it's even possible to thread them one inside the other to form a knot.